Hello, in the last lecture we looked at the first part of glycolysis, and today we're going to explore the second part of glycolysis. Just to reiterate, the first part that we've discussed already included the process of um, converting glucose through a series of enzymatic reactions into glyceraldehyde 3-phosphate and dihydroxyacetone phosphate. Now, as you remember, only one of these actually goes into the second part of hydrolysis, which is glyceraldehyde 3-phosphate. And actually, we have two molecules of glyceraldehyde 2-phosphate because the second one comes from dihydroxyacetone, which is also converted into glyceraldehyde 3-phosphate. So now let's look at enzymatic reactions in a payoff phase of glycolysis, which includes the steps 6, 7, 8, 9, and 10. Now, the first reaction in the payoff phase of glycolysis is catalyzed by glyceraldehyde 3-phosphate dehydrogenase, in which we add an inorganic phosphate to glyceraldehyde 3-phosphate. Now, please keep in mind that this is an inorganic phosphate rather than the phosphate from ATP. So all this reaction does is basically we take this hydrogen and substitute that with a phosphate. Now, funny thing is, we have this hydrogen being released, right? And then we have this hydrogen also being released. So we have net gain of two hydrogens. Well, those two hydrogens, one goes over here, and the other hydrogen is this. So we produce the NADH plus a proton here. And also we produce 1,3-bisphosphoglycerate. Now, of course, we will look at this reaction in a bit more detail. Just to mention again, the delta G prime 0 is 6.3. This is a reversible reaction. These products are being used downstream. Therefore, the product of this reaction is kept at lower concentration, driving the equilibrium towards the product. Now, this catalytic reaction is quite lengthy and has 5 distinct steps. So let's chop this down to individual steps and look at them one by one. So here's the first step of this enzymatic reaction. We are looking at the catalytic site of glyceraldehyde 3-phosphate dehydrogenase. Now, and this describes the formation of our enzyme substrate complex. So when we have NAD plus within our catalytic site, the whole site becomes more reactive. The pKa is lower. So that forms a reactive thiolate form. So having a reactive thiolate is basically another way of saying it has a negatively charged sulfur, right? Thiolate by definition is this thiolate. So when glyceraldehyde 3-phosphate comes into the enzymatic site, the carbon in a carbonyl group here makes a covalent bond by by sulfur attacking carbon and releasing an electron here to oxygen. So now we have a covalently bound substrate to the cysteine residue by a thiohemiacetal bond linkage, which is this right here, this linkage right here. So, as such, our molecule is primed for further enzymatic reactions. And in this case, what happens is we remember we had the release of an electron from carbon to this oxygen when this bond was made. Now this electron goes back to make a double bond, but instead of breaking this, the thiohemiacetal bond, it actually breaks this carbon-hydrogen bond. As a result, this hydrogen is taken up by NAD, and we form NADH, and this double bond, carbon double bond oxygen, is reformed. So now I hope you remember from the overall diagram of glycolysis. At step 6, we actually generate 2 NADHs plus a hydrogen, and we take up 2 NAD pluses and 2 inorganic phosphates. Now these are inorganic phosphates, okay? These are not phosphates from ATP. That's important to remember. So from the previous step, we had this in our enzymatic site. So now we have NADH that is ready to leave, and it leaves. What comes in instead of this NADH is NAD plus and another inorganic phosphate. So inorganic phosphate having a nice negative charge here readily makes a bond with our carbon of interest, and this bond actually is strong enough to break this carbon sulfate bond. So after breakage of this bond, we have generated our 1,3-bisphosphoglycerate, and remember we took in inorganic phosphate and NAD plus. So we basically regenerate the empty and ready to go glyceraldehyde 3-phosphate dehydrogenase. Okay, so a net result is taking up of one inorganic phosphate, one NAD, creating NADH, and bringing in the next NAD plus into the site for the next catalytic activity. Now, again, we take in 2 phosphates, 2 NAD pluses to generate 2 NADHs. Why? Well, okay, here is a good question. Why? How do we generate how do we take up 2 inorganic phosphates, 2 NAD pluses, and how do we generate 2 NADHs? The next step is also a very interesting step, because this is where we start generating ATP. Now we have the 1,3-bisphosphoglycerate. This is an interesting molecule. It has a phosphate on it that can readily be given away. It has a pretty low delta G prime 0. So we have our 1,3-bisphosphoglycerate. We add adenosine diphosphate, and we generate 3-phosphoglycerate and ATP. So now let's look at what exactly happens on an enzymatic level. I apologize, your book does not go into the details of the enzymatic reaction of phosphoglycerate kinase. But basically what's happening here is that the kinase transfers the high energy phosphate group, just like I said in a previous slide, from the carboxyl group of 3,1,3-bisphosphoglycerate to ADP, effectively forming ATP and 3-phosphoglycerate. Now, this compound here has quite high, has a low delta G prime, so this reaction is quite favorable in generating ATP. The next step in glycolysis is the enzymatic reaction that takes 3-phosphoglycerate and transforms it into 2-phosphoglycerate. All it does is it takes this phosphate and put it on a second carbon from the third carbon. Now this enzymatic reaction has 3 major steps, and now let's look at them one by one. Now this is a very interesting reaction because in an enzymatic site of phosphoglycerate mutase we have 2 histidine residues, one of which carries the phosphate and the other one has 2 free valence electrons on a nitrogen. Again, just like most of the reactions in our glycolysis, this is a reversible reaction, and this is what happens. One of the histidines that has a nitrogen with 2 valence electrons actually takes away the hydrogen on carbon 2 OH. So that frees up oxygen to react with phosphate, breaking this bond. Okay, so with that we generate carbon oxygen bonded to a phosphate on a second carbon. Now, interestingly, now we have the original histidine that had a phosphate on it. Now it has the 2 valence electrons. Now the architecture of the enzymatic site is such that it can react with the next phosphate. Now this intermediary compound is known as 2,3-bisphosphoglycerate, which makes perfect sense. The second carbon, third carbon, both of them having a phosphate group. The next step would be the histidine 1 reacting with the phosphate and the oxygen on carbon 3 reacting with the hydrogen on a second histidine. So in the end, we have reformed the oxygen-hydrogen bond, and we have taken up the second phosphate by the first histidine. Now this is just an interesting fact. I mean, we did generate our 2-phosphoglycerate, but it was actually this phosphate was originally bound to this histidine. So this phosphate that now is on the histidine actually came from our original 3-phosphoglycerate. Just an interesting point to make. Next reaction is catalyzed by enolase, and that reaction forms water by releasing 1 hydrogen and OH from the 2-phosphoglycerate that generates the phosphoenol pyruvate. Now we already have pyruvate in there, which means we're coming uh, pretty close to the end of our glycolysis cycle. So now let's look at how this actually works. I hope your book does go into the details of the enzymatic activity, but if it doesn't, it's quite self-explanatory. We have hydrogen leaving one electron here, which then binds here, releasing OH with an electron. So let me write this down. So we have carbon, carbon. This is carbon 3. This is carbon 2, which had hydrogen. Now it has hydrogen plus, carbon minus, and we have HO that now left. We have HO minus, and technically we have carbon plus here, right? And of course this is highly unstable, so this electron would come here, make a double bond neutralizing this charge differences, which then forms that. Well, unfortunately, your book does not go into the enolase enzymatic activity. I suppose these are complex, which makes sense, but the point is that we have now generated phosphoenol pyruvate, and phosphoenol pyruvate has a high energy phosphate sitting on it, and it can readily give it to ADP when it's catalyzed by pyruvate kinase in the presence of magnesium and potassium. As a result, we transfer the phosphate to ADP, to generate ATP, and because the phosphate leaves one extra electron here, here, that electron forms a double bond with this carbon, breaking the other double bond with a carbon between carbon 2 and 3, which then takes up the hydrogen that was basically released during this reaction. And the delta G of this reaction is negative 31.4 kilojoules per mole. Here I need to mention that the delta G of hydrolysis of phosphoenol pyruvate is closer to minus 61.9 kilojoules per mole, which is significantly more than needed to form the phosphoanhydride bond of ATP, phosphoanhydride bond right here, which is about minus 30.5 kilojoules per mole. That's why we have remaining delta G prime 0 of 31.4. Next, we have a quick little reaction that is catalyzed by tau isomerase, which is basically a transition of pyruvate from enol to ketone form, because the pyruvate first appears in an enol form and quickly tautomerized into a ketone form because that form is just more stable in a neutral pH. So it is much more favorable in this direction than in a reverse. Now that we know all the good that glycolysis or glucose does for us, now let's look at how some pathologies or pathological conditions take advantage of it. For example, cancer has very, very high glucose metabolism. Therefore, it is not unwise to think that sugar-free, or not sugar-free, but diet low in sugar is good for you. Nobody can refute that. What sugar is not good. We've been told that as kids, that it will rot our teeth. Anyways, it can do a lot more. So, glucose uptake in cancerous tissue is in order of 10 times faster than in normal, non-cancerous tissue. This is because, well, let's look at it from a perspective of a single tumor cell. Okay, it's cancerous. It starts growing. It's avascular, meaning it has no blood vessels. It has to survive somehow. It has to take up nutrients from its environment. It has little oxygen, right? So what does it do? It's in a hypoxia environment. So the anaerobic metabolism is the only thing that it can do to generate ATP by taking up more of glucose than normal cells. I mean, the cells need to have a certain amount of ATP. If they cannot do it through electron transport chain, then the cell has to go through anaerobic glycolysis, and that requires a lot more sugar. That also results in excessive production of pyruvate, and subsequently pyruvate is being turned into lactic acid to recycle NADH. So tumors have a hallmark of being both hypoxic and being very acidic. So hypoxic environment and what's called acidosis are very common features of a tumor's growth. In addition, to overcome some of these constraints that tumors have, some tumors overproduce several of the glycolysis enzymes. These include isozyme of hexokinase, that in particular cases associates with the cytosolic portion of the mitochondrial inner membrane. This particular isozyme is insensitive to the negative feedback loop from concentration of glucose 6-phosphate, so it's active. This enzyme can be synthesized to that extent, to such extent to overshadow the ATP production in mitochondria. So using basically converting the glucose to glucose 6-phosphate and continuing the glycolysis. No feedback regulation. In addition, hypoxia, as we said, that tumors do not have adequate supply of oxygen. Hypoxia also increases the prevalence of a transcription factor known as hypoxia-inducible transcription factor. This transcription factor starts transcribing mRNA that's to stimulate the synthesis of at least 8 of the glycolytic enzymes. All in all, this gives the tumor cells the ability to survive anaerobic conditions long enough for blood vessels to infiltrate into the growing tumor. Now, interestingly, also, hypoxia and acidosis are also triggers for blood vessels to start growing towards the localized area of hypoxia. The process is known as angiogenesis. It is the same mechanism that is used in the normal tissues to repair damage. It is the same mechanism that is used by um, developing embryos to arrange the blood vessels perfectly. So tumor cells take very good advantage of many of these mechanisms to survive hostile environment and then in the end take over the host. It was the German biochemist Otto Wennberg that was first to describe these events in 1928. He was the one to recognize that tumors have higher rate of glucose metabolism than other tissues. He was the one that was able to purify, crystallize, and purify 7 out of 10 of the enzymes in glycolysis. He was the one to come up with a method to uh, be able to assess directly the extent of glucose metabolism. And he was one of the most prominent figures in biochemistry at the beginning of the 20th century. Now to go back to biochemistry um, and continue talking about glycolysis. By now we know very well that the main player in glycolysis is glucose as the feeder molecule into the glycolysis cycle. But besides glucose, we have many other sources of sugars that can be used to feed the glycolysis. These sugars can include complex molecules such as glycogen and starch, um, smaller molecules such as galactose and mannose, fructose, sucrose. Um, of course, the most significant ones are as storage polysaccharides are glycogen and starch. The disaccharides such as maltose, lactose, trehalose, sucrose, and we have also monosaccharides such as fructose, mannose, and galactose. As you can see in this scheme, we have many different enzymes that are involved in transforming one type of sugar into another. And importantly, let's say we have glycogen and starch. Glycogen and starch can be converted into glucose by addition of water with the help of alpha amylase. And then, of course, glucose then is used, jump starts the glycolysis by being transformed into a glucose 6-phosphate through hexokinase, an addition of ATP. But also starch and glucose can also be used or hydrolyzed directly, rather phosphorylated directly into glucose 1-phosphate and then downstream into glucose 6-phosphate. Molecules such as galactose can also fit in through the same pathway. But fructose, for example, can fit in such a way to generate glyceraldehyde 3-phosphate directly. Well, so even though glucose is our main player here, it is not the only player. We rely heavily on many different types of sugars to promote synthesis of ATP and sustaining the glycolysis cycle. Here we have seen this already, so we will go over it briefly. Glycogen or starch. We have a terminal sugar, glucose, that can be a phosphate can be added to it or being phosphorylated. And basically, this event breaks the bond here we generate a glucose 1 phosphate, and we have our glycogen, whatever that chain is, it's a long chain of sugars with N minus 1. And glucose 1 phosphate is later used in glycolysis to generate ATP and pyruvate and so on and so forth. Now this is it for today. We will pick up next time by looking at the fate of pyruvate under anaerobic conditions. Basically, we will look in depth at fermentation.